Welcome to the pre-lecture for the BNG426 526 Metabolic Engineering flipped classroom activity. We are going to discuss some key points for the upcoming flipped classroom here. So first I will start by talking about facilitated transport. We discussed transport reactions early on in our metabolism warm-up sections lectures here. Facilitated transport, a carrier will bind the substrate in question. It'll form a complex and then it will deposit that substrate in the cell cytoplasm or cytosol, depending on what kind of organism you are. In the schematic here, we see an abiotic phase and a cytosolic phase. Essentially the carrier, which is this dual triangle here, will bind the substrate in that abiotic phase, change conformation, and then begin to unbind as the substrate approaches the cytosolic side, and then the carrier is able to deposit the substrate in the cytoplasm of the cell. And that has kinetics that look a lot like this equation down here, where you have the carrier plus substrate yields a carrier substrate complex, and this is a carrier substrate complex that is facing the abiotic phase, which then turns into a carrier substrate complex facing the cytosolic side, and then once again we are left with a carrier that's empty, and the solute or the substrate is now present in the cytosolic side. Passive transport is a little bit different from this. Obviously, because it does not need a protein facilitator. There's no protein facilitator, but the principle of down the gradient still applies. You're going down the concentration gradient. You're not going against the gradient, otherwise passive transport would not happen. Typically the solvent must be hydrophobic, the solute rather must be hydrophobic or small enough to traverse the cell's membrane bilayer. But we have basically already talked about this in class, so you should already know this. It's gradients like these that are responsible for making ATP in the cell. You've seen the F1, F0 ATP synthase here. Essentially this is run by using a proton gradient. Protons or hydrogen atoms, ions, are transported down the gradient in cells from outside to inside, and the gradient is produced in the first place by electron transport chains, and the transport of the protons down that gradient is what makes the energy in the form of ATP. So this brings us to talking about pH and acids, namely amino acids to start with anyway. And this will all make sense and it'll all be tied together a little bit later here. But for amino acids, when dissolved in water, they don't exist in the neutral form. So they have a certain charge to them usually. And that is determined by this thing called the pKa. And the pKa of a carboxylic acid group on an amino acid is usually around 2, and the pKb of the amine group on an amino acid is usually around 9.5. So at a pH of 7, so if we dissolve water, amino acids in water, the amino acid exists as what's called the zwitterion, which has both a positive and a negative charge on the same molecule. And you can see a schematic of a general zwitterionic amino acid with the positively charged amine group and the negatively charged carboxylic acid group. What does an amino acid look like at pH 1? Well, it looks a lot like this up here, again with the positively charged amine group, but it has a neutrally charged carboxylic acid group. That's because it's at a very low acidic pH with plenty of protons inside the area of note. What does an amino acid look like at pH 14? Well, we have a neutral charged amine group, but a negatively charged carboxylic acid group. But at pH 7, which is what we were just talking about, we have the zwitterionic amino acid here, where we have a negatively charged carboxylic acid group and a positively charged amine group. And why is this important? Again, we are going to see very soon. Namely the next slide. And what we really are caring about here in terms of this flipped classroom activity is the acid part of that equation. Specifically, we're talking about phenylacetate or phenylacetic acid. And you can see here structures of phenylacetate, that is the negatively charged carboxylic acid group, is called phenylacetate. But if it has a protonated carboxylic acid group, it will be termed phenylacetic acid. So that's the main difference here. But basically, phenylacetate or phenylacetic acid is the product of phenylalanine, which is an amino acid, the degradation of that amino acid, which is also a degradation component of lignin. So phenylacetate is a degradation component that can be formed if microbes are degrading lignin, which is a good component of wood. It is also a degradation component of styrene and ethylbenzene. Both of these are contaminants. So organic acids can potentially diffuse across the plasma membrane in passive transport if a gradient is possible. So both dissociated and undissociated acids can cross the plasma membrane by diffusion. However, the undissociated acid has a much larger solubility in the membrane and can traverse the membrane much more rapidly, as can be shown in the figure below, because it is not negatively charged. The negatively charged group, in this case the carboxylic acid group, adds a bit of a stumbling block to the diffusion of the organic acid across the membrane. However, once in the membrane, the organic acid will likely dissociate because the cellular pH is around 7.0. Carboxylic acids, as we've already established, have a negative charge of pH 7.0, and they would be called carboxylates. So we see here phenylacetic acid crossing the membrane, ending up in the cytoplasm, and then it will rapidly dissociate to phenylacetate plus a proton here. So this leaves a free proton floating around, which in turn acidifies the cytoplasm. So again, this means two things. Acids can get into the cytoplasm, but they will dissociate and then they will have a harder time getting out. Which, if you're a cell and you're using these acids as food, that's a good thing. That means they're not going to escape out the way they came in. However, the cytoplasm itself has a proton floating around, and overall the cytoplasm must avoid getting acidified. So this is not such a good thing if you're a cell, to have an acidified or have the threat of an acidified cytoplasm. You've got to balance that. So what a cell can do to balance the acidification, the pH of the cytoplasm in the event of possible acidification, is to reverse the proton gradient. So essentially, the F1, F0 ATPase, which in the proper direction, when it pumps protons back into the cell, going down the gradient, will make ATP. But if we reverse it and use it to pump protons out of the cell, we then have to spend ATP. So protons must be pumped out. The F1, F0 ATP synthase does this. It acts in reverse, thus spending a lot of ATP. So you can imagine organic acids, if they were to come in freely through just diffusing into the membrane, would end up acidifying the cytoplasm and thus not be the best carbon sources to be used for microbial growth. So the assignment here is to design an organism that has the phenylacetic acid degradation pathway in it, and you will see some readings associated with this assignment, and you should look at those readings. The cell should be able to eat phenylacetic acid. The phenylacetic acid degradation pathway is known, and a couple of the readings that you have will show a pathway of phenylacetic acid degradation. So in degrading the phenylacetic acid pathway, it should provide the cell with ATP. Consider what we were talking about here with the phenylacetate and phenylacetic acid transport into the microbial cell, specifically the diffusion through the membrane, and consider how the pathway itself, the degradation pathway, will hook up into the cell's metabolism. What key central metabolites are made by degrading phenylacetic acid, which you will see in the readings again. So the TCA cycle, here is a view of the TCA cycle. Again, we've already seen this. A lot of pathways can feed into the TCA cycle because the degradation of a lot of molecules end up making a lot of central metabolites. Often things like pyruvate, acetyl-CoA, oxaloacetate, fumarate, malate, perhaps succinate, even succinyl-CoA. These things can be made by degradation of certain carbon sources. Heterologous pathways, for whatever organism of choice is involved, will also potentially make central metabolites upon being degraded. So they can feed into the central metabolism, into the TCA cycle of a heterologous organism. And we will discuss that and see how to do that on Wednesday. So to do, watch this small recorded lecture. Presumably you have already done that if you are listening to me telling you this right now. Read through the 3 papers posted on my courses. Focus not necessarily on the nitty-gritty details, focus on the pathway itself. The results of the characterization of the pathway are what are important here, not necessarily the methods. So please don't spend too much time on the methods unless you really want to. What I would suggest is to read the abstracts and the introductions, study the pathway diagrams, and look at other figures to examine key results. But do not read from beginning to end of these papers. You will spend far too much time in preparation. Just focus on the pathway details and not necessarily the methods, as I said. And have access to these materials from my courses in class on Wednesday. Not necessarily this lecture. Perhaps the slides involved you could have access to. We will split into 5 groups in class on Wednesday. There is a student groupings file. It's a Word file with the online class materials for this stuff. So please look at that, see what number you are, and if you're a number 1, you'll be in a group with all the other number 1 students, and number 2 with all the other number 2s, etc., etc. You know how it works. I will provide a handout in class. I'll print it and bring it to class, answer the questions on the handout, and at the end of class I will collect these, regardless of how far we've come, submit it at the end of the class for credit. And again, like I said at the beginning, this is really about effort, not necessarily about getting the correct answer or something. In this case, this is working in a group. Let's work in a group and do it effectively. So as long as you can do that, you will be okay. Don't worry if you're leaving questions blank or haven't quite finished everything by the time 150 rolls around during class on Wednesday. Just try your best at it. Again, for all I know, and I've mentioned this already, for all I know, this could take only 10 minutes. Maybe even after 2 classes we won't finish it. Who knows? But we're just going to kind of gauge how it goes and just try your best, and hopefully you will enjoy the process. So good luck, and if you have any questions, send me an email or talk to me before class on Wednesday, and I look forward to trying this with you. Thank you.